Dr. Richard Hayward, who is a Royal Society Research Fellow in the Institute of Structural and Molecular Biology. The Institute of Structural and Molecular Biology is a joint initiative between Birkbeck and UCL, and many of the recent appointments in biological sciences have been made jointly between the two institutions. So Richard's been at Birkbeck for about 4 years as a Royal Society Research Fellow, having come from Cambridge, where he was for the previous 13 years, but originally he was an undergraduate, I think, in York. And he's going to be talking to us on sex, drugs and rock and roll, chlamydia, unlocking the secrets of a stealth pathogen. Thank you.
Thanks very much, Nick. Good evening. It's nice to see everyone. As Nick said, I've been fortunate enough to be supported as a Royal Society Fellow for the last 7 years, and I was able to transfer that funding here, where I started my own research group in microbiology. So hopefully I'm going to cover a lot of bases in terms of level to make sure that some of this talk is understandable and that people will take away something for themselves regardless of your prior knowledge. So for those who have more knowledge, you'll find that as the talk goes on, things get slightly more complex, so please bear with me with that. So I've been a microbiologist for the vast majority of my research career. And this involves the study, by definition, of very small organisms, be them bacteria, viruses, or parasites. And so by definition, we can't see these organisms by eye. And just to give you an idea of scale, we're down here in the micron to submicron range when we're thinking about the bacteria. And way down here in the nanometer range, we have viruses, going down to much smaller proteins and eventually atoms right down at the bottom of the screen here. And so our observational basis for biology relies a lot on light and electron microscopy so that we can observe these organisms and the way in which they interact with each other, and in terms of disease, the host. And we can also look at the proteins these bacteria deploy in terms of their ability to cause disease. And this has really been the basis of my research over a number of years, trying to understand the molecular basis for the interaction between pathogenic or disease-causing bacteria and their higher organism hosts, humans and animals. And so just here to remind some, and probably as information for others, I have a generalized structure of your typical bacterium. So essentially a bacterium is bounded by two membranes which are made up of lipids or fats, and within that is enclosed its genome made up of DNA, which is loosely floating in the cytosol, which is essentially the reaction mixture where all the metabolic reactions take place within the bacterium. Some bacteria are able to move around under their own power using this organelle on the surface called the flagellum, which is essentially a molecular motor and has all the facets you might expect from an engine. And also from the perspective of avoiding recognition by the host, there are a lot of surface structures on these bacteria that they use to stick to surfaces and to avoid being eaten by cells of the immune system. And these are both sugars, carbohydrates, and proteins. And they must vary these structures on the surface to avoid detection. So there are a lot of genetic mechanisms that underlie these processes. And it's really through studying these humble bacteria we now have an appreciation of the genetics of higher organisms. So bacteria can be mutated very easily, and we can, by mutating one particular gene in their genome, we can interrogate the function of the particular protein which is produced. And of course, this is a very generalized schematic, and we should remember that bacteria, like us, I guess, come in many shapes and sizes and forms, but most will adopt this generalized structure. So as we go through the talk, we just have to remember that the bacteria essentially have a genome, have two membranes which are at the boundary of the structure, and within there is the cytosol. And in the media, we tend to come across a lot of bad press for poor bacteria. And of course, it's important to remember and to be aware of these problems that we have in our hospitals, like MRSA, and these food poisoning outbreaks, which we occasionally hear of, our E. coli 0157 outbreaks in our hamburgers. But I think this gives us a skewed perspective of the extent to which bacteria are actually responsible for causing disease in the human population. So I posed a question down here, and I think it's important to remember in the context of this talk and my research that not all bacteria cause disease. And so how many bacteria are actually causing disease for a lifetime? So I've called this slide the good, the can be bad, and the ugly. And really you can think of it as a pyramid where we have the most numerous bacteria sitting at the top here, and we have the fewest bacteria down in the bottom category in red. And what we see is that the vast majority of bacteria are the angelic bacteria on the right-hand side here, the so-called commensal or good bacteria, which never cause disease, and they can actually do helpful things for you. There are more of them in your body than your own cells, and so you're actually more bacterian than you are human. And they're actually doing helpful things. So they're protecting the surfaces of your body from incoming invaders. They're doing helpful metabolic reactions in your intestine, and even E. coli, which gets the bad hamburger press, in its more benign form, is doing very useful things in your own gut, and without them your metabolism would be much the poorer. So actually these good bacteria are the more numerous and yet the very much understudied and well populated in terms of the general press. As we come down, we have groups of microbes which are potentially pathogenic, which are in the wrong place at the wrong time. And we might think here of gut microbes which inadvertently get into the spinal fluid, disseminate into the brain and cause meningitis. But that's not because their job in life is to do so, but because they're essentially in the wrong place at the wrong time. We think also of things that hit the news like flesh-eating bacteria. These form of staphylococcus which invade into your skin through wounds and then have these quite dramatic effects. They're not naturally adapted or evolved to do that, they're just in the wrong place again at the wrong time. And there are various cousins of the humble E. coli from up here, which by gaining a few extra genes actually turns them into something which causes diarrheal disease or urinary tract infections. And then down here, where we're going to concentrate for the rest of the talk this evening, are those pathogens which you would argue are possibly not as well developed as these ones at the top here, because when they interact with the host, they actually cause a disease to you. And on an evolutionary term, you can imagine that these are possibly less well adapted because the host works out that they are present, and some of the responses that the host makes causes you to engender a disease, whereas these more numerous ones at the top are actually interacting you on a daily basis and essentially are doing so silently. So if you think of that in terms of evolution, you could say that these non-pathogenic bacteria, which has become quite a central focus of current research in the last couple of years, are actually much more well adapted to that interaction than those we spend the vast majority of our time studying. And so, of course, why is it important to do this? Well, the new chief scientific officer for the government reminded us the other day that antibiotic resistance will become is as big a risk as terrorism to the UK government, unless when you look at the funding that is put into these microbes and this problem compared to other policy, we find that there's a vast discrepancy. And this is because it's predicted that if we continue to use the antibiotics that we use to combat these bacterial diseases that we've been using for a number of decades now, this is exerting and continues to exert such a selective pressure on these organisms that they are, in terms, engaging in an arms race with us, such that they're continuously developing ways to circumvent the drugs that we're using against them. And this is becoming at such a precipitous point now that it's predicted within the next decade or two, these long-souled and long-fangled weapons coming from fungal sources will no longer be useful against even the most common threats. And so if you're unlucky enough after surgery to get an infection with a common microbe in a decade's time, it may not be as easy to combat that as it might perhaps seem today. And so we must plan for the future, and we must think about, instead of using this chemical weaponry that we've been throwing at bacteria for decades, how we might perhaps intervene in different ways. And it's through studying the molecular biology of these pathogens, through the knowledge of genetics, that we might be able to do this in a more directed and precise way in the future. And so the drive of understanding these host-pathogen interactions is to examine their mechanisms on a fundamental level, which is what we do in my lab, in the hope that we might be able to use that information in the future to develop new modes to combat bacterial infection. So thinking more about those host-pathogen interactions, these pathogenic bacteria interact with their higher eukaryotic host, which could be us or a chimpanzee, at a cellular level. And this is just to remind you that in higher eukaryotes, cells come down here. These are the fundamental building blocks of tissues And it's these tissues in turns or accumulations of macromolecular architectures of cells which come together to form our organs, in turn our organ systems, and make up an organism. So from the perspective of what we actually study in my lab in terms of the chlamydia host interaction, we're interested in understanding how individual bacteria are interacting at a molecular level with these fundamental building blocks of our tissues in the host organism. Now, cell biology is quite complicated and full of acronyms. And so perhaps for a non-specialist, this can be quite daunting to understand. So here I have a schematic diagram of the fundamental units that go up to make one of these higher eukaryotic cells. And we see that there are a number of components within this boundary membrane. And I think the easiest conceptual way of thinking about this, if you're not familiar, is to think of this as analogous to a city. So we have in the center the nucleus, which acts as the brain or the control center for the activities of the cell through the control of the chromosomal activities and gene expression within the eukaryotic cell. And this is bounded by a membrane, unlike in the bacteria. The cell is then bounded by a single membrane, which we call the cell membrane or plasma membrane, which acts like a fence to keep things out, but also must be permeable in the sense of border control to allow things in and out. Within this membranous bag, we have a cytoplasm, just like in the bacteria, where reactions occur. You can imagine people meeting, or perhaps because it's so crowded, it's more like a nightclub. And we have a number of membrane-bound organelles within here, and it's these that we're interested in terms of the molecular biology and how the interactions are controlled. And these are organelles which perform particular jobs which make sure that the cell functions. So, for example, we have this structure here near the nucleus called the endoplasmic reticulum, which is essentially a protein-making factory. And this is studied with small structures called ribosomes, which are the units responsible for making protein out of genetic material. We have the Golgi apparatus, which is essentially a storage organelle. So here we have a warehouse. And we have a number of smaller organelles, such as the mitochondria, responsible for energy supply, the lysosomes, which are responsible for degradation. And within these vacuolar compartments, we also have a police force, a repair system, and transport mechanisms. So you see the cell is built up fundamentally of the same things that we see in life, but we're just giving them these complex names. So we'll come across these names during the course of the lecture, so I just thought it was important to have a grasp of the fundamentals of this in an easier, understandable way. So in the mid-90s, about the time I entered the field as a PhD student, this was the dawn of a really exciting time in microbiology. Prior to that, people had been characterizing and categorizing organisms and trying to understand how they were related to one another. But at that time in the States, there were a number of groundbreaking experiments, possibly done by accident in different laboratories, where people added different pathogenic bacteria to their cultures of eukaryotic cells. So here we have under the microscope one of those cells I showed you in the last diagram. Here is the boundary membrane, here is the nucleus, and here we don't see very well would be the other organelles. And within here you see there are listeria bacteria actually flying around. On the bottom here we see that if we put salmonella into these cultures, they will induce all sorts of changes in the cell membranes, and this will lead to the uptake of the bacteria inside the cells. So this was really a revelation. Instead of using agar plates and just growing these bacteria in isolation, if you like trying to categorize them, we could now ask more fundamental questions without the use of animals about the molecular mechanisms by which they were causing disease. Because we can combine different sorts of bacteria mutated in different genes, and we can add those to cells, but we can also interfere with the cells. So we can perturb the function of the nucleus, or we could perturb the function of some of these intracellular organelles, and then ask what happens to these kinds of processes. What you'll also notice is that the bacteria obviously have very much more knowledge of this than we do. So they've been reading Watson's book of the molecular cell biology very thoroughly, far in advance of us, and they're very adept at knowing this roadmap of how to get around inside the city. And so by studying these bacteria and their weaponry, we can actually gain new insights into how our own cellular processes, which are important to understanding other things like cancer, are controlled. So if we think about what the weaponry may be by which these bacteria are causing disease. Here we have Q from 007, and we look at his arsenal of different weapons, and we realize that bacteria are causing disease by using different weapons in their engagement with these eukaryotic cells. And probably up until the 1980s, all we really knew was that some pathogenic bacteria were secreting molecules called toxins, which were able to interact with the host cell down here. And these were able to engage with the host cell in two different ways, either by inserting into that boundary membrane and creating a gate so that the host cell was blown open. And we think in this case of things of hemolysins and other things that are able to lyse red blood cells from E. coli. And also, we have a group of toxins called AB toxins, which were able to enzymatically poison the cell once they get inside. But the net result of both of these, whether it be cholera toxin or anthrax toxin, was basically to destroy this host cell. Now, if you're the bacteria and you're trying to interact with the host, what you really don't want to do is carpet bomb the epithelial cells that you're trying to interact with. And essentially, that's what these weapons do. These are essentially weapons of mass destruction. They can be useful in terms of preventing recognition by the host immune system. But when we look at such sophisticated movies as this, we see that the bacteria are not actually killing the cells, but exquisitely subverting them for their own purposes. And through the sequencing of bacterial genomes at about the same time, we realized that there was a new common weapon system that was present in a lot more of these pathogenic bacteria, that was a much more sophisticated and stealthy weapon. So here we align it to a stealth bomber. And this had a conserved structure. So instead of just secreting toxins, which then interacted with the cells, these systems were essentially a syringe-like structure, which were built across both the bacterial membranes that we remember from the first slide, traverse the space in between, interact with this eukaryotic membrane, and were then pumping, just like a hypodermic syringe, what we call translocated effectors into the cells. And so these more complex interactions, it seemed, were being governed by these more complex systems. And if we think of some of the well-known viruses, it's been known for 20 years that viruses were very complex in their interactions. They were certainly not blowing holes in cells. And now this kind of aligned the story to suggest that common bacterial pathogens were actually engaging in very, very sophisticated crosstalk between the pathogen and the host cell. And when these structures were isolated for the first time and visualized by electron microscopy, so here we have some of these injectosomes, as they've been called, that have been isolated from the membranes of Salmonella, we see that they do indeed resemble a hypodermic syringe. So they have two rings which engage with both of the bacterial membranes, and a protrusion which has been called the needle, which reaches out to the host cell. And many, many labs around the world have been studying this delivery apparatus in quite a lot of detail. And now, through reconstruction from electron microscope images, we have quite a lot of insights into what this structure may look like. And why is this attractive? Well, it turns out the more we look, the more we find it. And so if we're looking for a common new target for an antimicrobial in the future, we might imagine that some directed inhibitor of these injection systems might be a very pan-pathogen inhibitor, because we find them in diarrheal pathogens like Salmonella, in the plague-forming bacteria Yersinia, where it paralyzes macrophages through injection of proteins, and even in E. coli 0157, which sticks to the outside of your cells. The E. coli is actually sticking through the action of delivery of multiple proteins through one of these injection systems. And as we'll come on to, chlamydia, which we now study in my lab here, also has one of these type 3 or injection systems, which is responsible for the delivery of proteins across eukaryotic membranes. And we see from this that the effectors, or the cargo that is delivered through these systems, can have quite different effects. So although this structure that is doing the delivery is conserved, what comes through it is subtly different. So here, for instance, the Salmonella is using it to induce its own uptake into the cells, whereas Yersinia is using it to prevent the immune system from taking it up inside the cells. And we can even find examples in chlamydia and salmonella where the membrane is not the boundary membrane, but it's actually the internal membrane of a vacuolar or a membrane-bound compartment inside the cell where the bacteria are surviving and replicating. So this seems to be a conserved machinery for the delivery of these toxin or effector molecules in lots of different contexts in these bacterial pathogens. And we can even visualize this now using fluorescent tagging. So here we see the cargo in red and the bacterial membrane in blue, just like the schema here. And we see that there are various stages where we can visualize the cargo inside the bacteria and then its injection inside the host cell until the cargo is completely lost from the inside of the bacteria. So this is indeed injecting these effector molecules directly inside the host cells. And as I alluded to, this is not only interesting in terms of understanding how we might combat these diseases, like salmonella and E. coli and chlamydia, and to an extent mycobacteria, but also it gives us a lot of new insights into how different processes in our complex eukaryotic cell, our city, are controlled at a molecular level. And through studying these pathogens, we've actually got new insights into the function of our own cells. So to give you an example, you can see here that both E. coli and salmonella are manipulating the structure of the surface of the cell. And they do this through changing the underlying so-called cytoskeleton, which is like a scaffold which holds the structure of the cell together. And in this case, we see that it induces the movement of the cell in this direction to take up the bacterium, whereas here E. coli is making a structure from the outside to which it sticks. Now, if we align that to what we know about what cells normally do, we find that mechanisms that the bacteria trigger here are very similar to those the cell would normally use to move. And the mechanisms triggered by E. coli here are very similar to the mechanisms we know that cells use to stick. And what's significant about that is that when these processes go wrong, this leads to processes like carcinogenesis, tumor migration, and tumor generation. And so through studying the mechanism these bacteria are using to change the host cell structure, we can actually understand more about how cancer and metastasis is generated in higher eukaryotic systems. So even eukaryotic or higher organism biologists have suddenly become interested in our humble bacteria because they realize that by understanding their mechanisms, we can actually have new insights into both the control of cells and what happens when those control mechanisms go wrong in cases of cancer. So that was really a preamble in the hope that that will introduce you to what I'm going to talk about in more detail now, which is more focused on a single system and our research here at Birkbeck and at UCL. So we're studying the obligate intracellular pathogen, chlamydia. Now, obligate intracellular means that the bacteria has to survive within a eukaryotic cell. There's no way chlamydia can actually be free living on one of these agar plates we saw in the previous slide. So it's much trickier to study. And yet it has worldwide medical impact for two reasons. Firstly, and probably more well known, it's the leading bacterial cause of sexually transmitted infections in the Western world and in fact worldwide. And this is through the interaction of the bacteria with the epithelial cells of the genital tract. And this leads to infertility and can lead to a syndrome in women called pelvic inflammatory disease. And you see on this X-ray here that the tubules of the upper female reproductive system have become inflamed and swollen as a result of the interaction with Chlamydia trachomatis. And in addition to this, we know that prior infection with chlamydia predisposes at least women, and we'll come on to men later in the talk, to a possible higher risk of subsequent cervical cancer. So there's an interest both from a sexually transmitted infection standpoint and also this later association with a higher rate of cancer incidence in women prior infected with chlamydia. Perhaps less well known, or less well talked about, is the fact that chlamydia can also infect in exactly the same way. Instead of the cells of the genital tract, the cells of the inside of the eyelid. And this leads to inflammation of the inside of the eyelid through the occupancy and damage of the cells by the chlamydia. which leads to irritation of the inside of the eyelid and the scarring of the surface of the eye. And this leads to a form of blindness very specific to chlamydia called trachoma. And this has been recognized as a neglected tropical disease by the World Health Organization. And although we can treat this with antibiotics, this form of blindness, this is obviously in light of the fact that these bacteria are developing more and more resistance to these drugs, potentially going to be a problem in the future. From the standpoint of the sexually transmitted infections, most of these infections are actually asymptomatic. So if you think about a lecture hall, the current figures for 16 to 24-year-olds are that there is something like an average of 8% infection rate. So that means that all these green people in this lecture theatre would be carriers of chlamydia in an unknowing way, and actually it would be only the blue person who would know that they needed treatment through antibiotics through visiting the GP. And so the problem is there's a large resident, mainly predominantly young population who are carriers of this disease. And although this can be treated with antibiotics, there's currently no vaccine available. And it's very hard to control the spread because most of the people who have this are asymptomatic or show no symptoms. So from a disease burden perspective, we can see that chlamydia is of major importance both to the UK population and also worldwide health. So, how do we study chlamydia in our lab? As I said, it's an experimental challenge because there are no ways of growing this independently of eukaryotic cells or higher cells. So we spend a long time, instead of looking at agar plates, as some of you may be familiar with, on which you normally, the nutrient media, you grow bacteria, we have to grow these in cultured higher organism cells, so cell lines that have been isolated from human tissue or animal tissue. There is also no way of genetically manipulating chlamydia, so unlike E. coli, we can't mutate any of its genes and find out what it does. And for a long time, and it's perhaps surprising that even 10 years ago there was still a debate, there was a lot of consternation about whether chlamydia was actually a bacterium, a virus, or a parasite, and we'll come on to the reasons for that in a minute. So at the bottom here, we have the life cycle of chlamydia, which we can reconstitute in these cultured cells in the lab. And chlamydia is atypical, and this is one of the reasons that there was all this controversy, because it exists in two different forms that are actually the same organism. So this so-called EB, or elementary body, is able to attach. So here we have the host cell in red, and the chlamydia stained in green by fluorescence microscopy. And here we have an electron micrograph of the same interaction between the cell and the bacterium. And we see that these elementary bodies are very, very small. They're only around 300 nanometers in diameter, so about 4 times smaller than E. coli. And when they essentially can't divide or grow, but when they hit a cell, they will incredibly actively internalize. So they will coerce this cell, which normally doesn't eat things for a living, to take them up into a vacuole and eat them and bring them inside. They will all coordinately migrate to the center of the cell, where they will form this specialized compartment we call an inclusion. So this is essentially a membranous bag built by the bacteria, which is completely separate from the workings of the cell itself. And they use this as a niche in which to begin their replication. And when they begin to replicate, they also diversify into a second form, and we call these reticulate bodies, or RBs, and we see them up here. So now we have the nucleus of the cell as the big black hole at the top, and we have, this is part of the eukaryotic cell, and here we have the bag containing the chlamydia, which we now see are much bigger than the chlamydia over here, and their structure by electron microscopy looks much, much more like your common bacteria, rather than this metabolically inactive, dense structure on the left-hand side. Eventually, these will redifferentiate, and the elementary bodies will be then released back out into the extracellular space to infect a new cell. And we can recapitulate this cycle in the lab through the cultured cell model, and this takes about 72 hours. So this gives us a very good experimental model for looking at the ways in which these bacteria are interacting with the host cell at different stages. And given its medical impact, it might surprise you to know that there are so many question marks on all these arrows. I didn't bother putting them all in, but essentially we don't understand how the bacteria get inside. We don't understand how they get to the middle of the cell. We don't understand how they differentiate or redifferentiate, and we really don't understand what's going on in terms of how they escape the cells. So in terms of open questions and ways in which we could intervene in these processes, there is still a lot of open science to be done. So to give you an idea of what we actually see, here is a cellular monolayer. So here we have multiple eukaryotic cells all sitting on a dish. These structures are the nuclei, but you can't see the boundaries very well because they're all sitting together. And what you see after a few hours, this is massively sped up, is the development of these white structures in the cells. And this is the inclusion, which is growing and growing, containing replicating bacteria you can see moving. And eventually we reach a point which is consistent with around 72 hours after infection, where this compartment takes up most of the cell, and eventually you see the cells burst and release new bacteria. So we're interested in how the chlamydia is doing this, how it's tricking the host cell into allowing it to reside inside, essentially silently. And we got some clues from the genome, which was sequenced back in 1998. This told us a number of things, that it had a relatively small genome, only nearly 900 genes. But surprisingly, through use of comparisons to other organisms, we could only guess the function of about 600 of these 900 genes, and that's unusual. So it left about 255, which didn't look like anything that you would find in a human or a mammal, or anything that you'd seen before in another bacteria. So this was quite surprising. Almost a third of the genome looked like something completely alien. The genome was reduced in the sense that this was probably an adaptation you see in other organisms that are well adapted to living inside cells. The reason for this, it doesn't need as many reactions to generate nutrients because it can scavenge things from the nutrient-rich eukaryotic or higher cell environment. There was no particular lineage relationship to other bacteria, and we even found that some of the genes were more similar to plants than animals. So it really raised the question that prior to the dinosaurs, what really did happen? Chlamydia was about 4 billion or so years old, so it's predicted. And so this was something that diverged away from other bacteria very, very early on in evolution, and is essentially developed, presumably through the activities of these unknown proteins, which look like nothing else, the ability to undergo this rather complicated and convoluted cycle, and yet the exquisite size inside cells. So we've been beginning to tackle both observationally and biochemically some of these questions. And this, first of all, required us to set up a robust system to cultivate and recapitulate this cycle. But once we've done that, we've got trying to tackle questions, and I'll show you some examples of some of the things that we're doing, hopefully as a taster for you to become more interested in the future. So the first thing that we're trying to do is understand more about this entry process. And together with collaborators, we were the first to show that the injection apparatus was actually active and transposing one protein which reorganizes the scaffolding or cytoskeletal system of the cell inside to promote this entry phenotype. But what was strange is that this entry event, when we started looking at it, wasn't quite like you might expect. So what we imagine from the cartoon is that the bacteria come in and hit the cell surface, and we see the cytoskeleton here. These are the girders that hold the cell in shape together, called the actin cytoskeleton. And we see that there are chlamydia that we've added adhering to this cell surface. But what we might expect from the cartoon is that there's a massive reorganization of this cytoskeleton and the membrane. It all happens very quickly and the bacteria come inside. But what we see if we look more carefully is that something more subtle is going on. That actually very defined, very thin structures are reaching out to these bacteria from the cell. So this red line reaching out to the green chlamydia. We see it again here, and we should be able to see it again up there, but can't. And we can see it over here in higher magnification, suggesting the cell is actually extending an arm and making some sort of contact with the bacteria that is then bringing it inside the cell. So there's no global reorganization going on at these points that you would imagine from these early cartoons. And so we were interested in looking at these events in higher resolution, and we're very fortunate at Birkbeck to have a world-leading electron microscopist, Professor Helen Sable. And so we've been working together with Helen's group to try and develop new ways of using electron microscopy to look at these host-pathogen interactions, but not as individual protein structure, but at the interaction between the bacteria and the host cell in higher resolution. So this is what you see if you look down the electron microscope at one of these samples I showed you by fluorescence microscopy on the previous slide. So here, just to orientate you, you have various pieces of that boundary membrane of the eukaryotic cell coming around here. And we have the cytoplasm of the eukaryotic cell, and you can just about make out these lines, which are components of the underlying cytoskeleton. And what we see here is there's a close contact between the bacteria here and the host membrane here. And that we can very nicely discern, in fact we can actually measure the diameter of the outer membrane of the chlamydia and the inner membrane of the chlamydia. And we can also see something that was kind of unexpected and new, suggesting there's another membrane here, which we're still trying to research and work out what this is. Anyway, that's all very well for looking at a flat section. What we've been trying to do now is to do electron microscopy in 3 dimensions. So this is a slightly different technique, and I've brought the fluffy chlamydia to try and help. So what this entails is, so the previous picture, we were essentially looking down on the top of the chlamydia, and all you see is its flat hairy head here. In terms of the 3-dimensional electron microscopy, what you do is take a series of different pictures, but you tilt your chlamydia at the same time. So you take a picture here, a picture here, a picture here, a picture here, and do the same in the other direction. And then using computational software, you take this tilt series, and you ask the computer to realign all these sections using some markers you put into your sample, and then reconstitute these images in 3 dimensions. So this leads to a 3-dimensional high-resolution view of these interactions. And through tracing, rather laboriously at the moment because it's not automated, all of the different structures in these different sections, we can actually build up 3-dimensional models of these interactions. And this gives us unprecedented detail to understand how this host cell is reaching out to contact the chlamydia. So just to orient you, we now have the outer membrane of the chlamydia in green, the inner membrane in blue, this extra thing we're trying to understand in yellow. And we see that there's an incredibly complex network of membranes. In fact, there's 3 different protrusions in blue, red, and purple that are all reaching out to contact this bacteria. And then it's contacting it across the whole surface at different depths. And it's then, if you like, lassoing it and bringing it inside the host cell. Now, this is in complete contravention to what really was imagined as a cartoon figure of what might be happening here, and suggests the underlying mechanisms are very different to what has been imagined previously. So that's one angle of what we're developing. Another and major focus really is understanding how this inclusion, this specialized intracellular compartment, develops at the other end of the life cycle. So this boundary membrane of the inclusion, the so-called inclusion membrane, now becomes the interface between the host and the pathogen. And we think that chlamydia is exquisitely controlling what is going in and out of this compartment, but we're yet to understand fully the molecular mechanisms by which it does this. On the one hand, it keeps the cell from recognizing the fact that the bacteria are present, but on the other hand, the bacteria have to eat, so they have to control the transport of nutrients into this compartment as well. So there must be very careful control of this boundary membrane. And so by using a raft of techniques, we're beginning to look at both the cell biology and the biochemistry of what happens at this membrane. So this is just to remind you of our friend the cell city, and to point out that the endoplasmic reticulum, which I'm about to talk about, is the protein factory in the eukaryotic cell, and it's abbreviated to ER. So in looking at these chlamydial structures, we've shown a new interaction that was previously unforeseen between this inclusion, which we now see by a different form of fluorescence microscopy called confocal microscopy, where we can stain cells. So here we have a red eukaryotic cell again, stained with a marker which is present in the endoplasmic reticulum. We have the cell nucleus, and we have the inclusion containing the bacteria around the edge. Now, the difference between fluorescence and confocal, a bit like the 3D EM, is that this is a laser-driven microscope, so you can actually ask it to take optical sections at different levels through your sample. So what we've got here is the top of the inclusion, and then we move the laser down and take another image, and move it down and take another image, and another and another and another, until we get to the bottom. So this allows you, instead of in the EM, but in the fluorescence image to build up a three-dimensional view of this object, and allows you, as well as a top view, to have a side view. And this tells us, as you can see, that this red staining is enriched very clearly at the edge of the inclusion, which was previously unforeseen. And hopefully you can also see that there are some dots of red that get actually inside the inclusion, suggesting that through interacting with this protein factory of the host cell, chlamydia is doing something really important to promote the generation of the inclusion. And by interfering with the function of the endoplasmic reticulum specifically, we can show if we inhibit its functions, this inclusion collapses. So this is very, very fundamental to the establishment of this compartment inside cells. And so to look at this in more detail, we went back to the electron microscope. And here again, just to orient you, we have the inclusion lumen of the inside. We have a bacterium here with its two membranes. We have the inclusion membrane here and the rest of the cell over here. This is in fact the Golgi, the warehouse storage system over here. And what we see in these two boxes is very clearly the recruitment of the endoplasmic reticulum, because we know that the endoplasmic reticulum contains those ribosomes which are responsible for making proteins in cells. And we see these as black dots. So you see there's a membrane all the way along the inclusion membrane, and on this side, this membrane contains black dots characteristic of the ER. So this correlates with what we've previously seen. And if we then do our three-dimensional reconstructions again, this steps through each one of those tilted images and then shows you what happens when we start to draw around. We can build up a model in 3 dimensions of this complex interaction going on at the inclusion membrane. So now we can see that the dark blue membrane here is the endoplasmic reticulum, and all those red dots are the cellular ribosomes. And we can see very clearly that it's very close to the light blue inclusion membrane, and that when the dark blue membrane on this side contacts a bacteria on the other, it'll stop spinning in a minute, we see that there's an assembly of something contacting the bacteria and this membrane here, these brown complexes. So we were very interested and excited in what these may be. So if we look in more detail, we actually see that these are representing complexes spanning the bacterial membrane. So here we have the bacterial inner, bacterial outer, something in between. We have the inclusion membrane here, and this is the endoplasmic reticulum that's come in on the other side. And if we use an antibody coupled to a gold bead, we can show that this labels these structures in the bacteria very nicely. And this antibody is raised against a component of the injection needle. So it suggests this injection system is being assembled at the point at which this rough endoplasmic reticulum contacts the inclusion membrane. And if we take a slice in the dimension into the board, we can see that there are not 4 complexes, as you might expect, but a whole raft that makes some sort of 2D array in the membrane. So this is a site at which the bacteria are actively injecting, you might believe, proteins across this membrane into the eukaryotic cell. And we see that these structures only form where we see endoplasmic reticulum on the other side. So where we see no membranes, we don't see any structures, or other membranes, we don't see any structures formed here. Whereas when we look at endoplasmic reticulum, we see very clearly these tooth-like complexes forming across the bacterial membranes, characteristic of these injection systems. And we can even model those structures I showed you on the first slide into the densities that we see in the electron micrograph. So this really gives us the first snapshot of these injection systems in action in any system. So this is quite exciting new data. So another focus of the group is to try and understand what comes through these injection systems. And we know that there are a lot of chlamydial proteins, probably 40 to 100, which are pumped into this inclusion membrane. And we propose that these are likely to interfere with host cell functions. Now, these proteins are difficult to handle, but we've managed to purify them to do some biochemistry for the first time through genetic modification of lab E. coli and being able to make them individually in a test tube. And this enables us to ask questions about what each one of these proteins may be binding to in the eukaryotic cell. We've also noticed that they're able to bind to one another, that some of them are actually forming holes in this inclusion membrane, which may be transporting things in and out, and others have quite a striking ability to take lipids which should come together in circles and make them into tubes. So this suggests that the insertion of these proteins into this membrane is actually sculpting the shape of this boundary membrane of the inclusion. And this is an ongoing set of work. This is quite complicated proteins to handle, but we're beginning to see what sort of functions may be important for the cargo which comes through this injection system that we see next to this ER membrane, which you might imagine over here. And just to give you one example of that, we can generate particular cell types of cells, of eukaryotic cells, which are able to express through genetic engineering individual substrates or inclusion proteins. And so we can see them, we fuse them to the jellyfish protein, green fluorescent protein, which means it lights up under the microscope. And we've recently discovered that one of these inclusion proteins, quite provocatively, is able to interfere with another scaffolding system present in the cell, that being the microtubule network. And this is very important for cell division. And you see when the GFP inclusion protein is present, all of the microtubule architecture in red here is messed up. You see it's all assembling in the wrong place, and that the filaments are very, very short in comparison to this cell over here in white, where that is a normal microtubule network visualized by the same technique. So this suggests one of these inclusion proteins, which is coming through this injection system, is able to interfere with this fundamental scaffolding system in cells, which is critical to their ability to divide. And not only that, it's one of the systems that goes wrong and can predispose you to cancer. So it may be the link, or one link, as to why prior infection with chlamydia and the delivery of these proteins could predispose you in later life to some defects in your cells, which leads to an increased likelihood that these cells may go wrong. So just in the last 5 minutes or so, I wanted to think more about how that fundamental and mechanistic and biochemical research might, and some of these imaging techniques, might yield some information in terms of the clinic. And one fact that caught our attention and spawned a collaboration with Adam Brooks Hospital in Cambridge and the Health Protection Agency arose from observations in the National Chlamydia Screening Programme. So that said that 1 in 10 women, but actually 1 in 7 men, were infected with chlamydia. And as we said before, we know very clearly that these effects and the infections in women lead to the pelvic inflammatory disease, and this can cause infertility and ectopic pregnancy, and also increase the risk of cancer. What we don't really know, and what isn't clear at all, is what happens in men. And actually, there are more men infected than women. So we know that some chlamydia can or are consistent with patients that have urinary tract infections and fertility problems. And we can show, others have shown, that experimentally, if you take chlamydia that you've grown in the lab and put them onto spermatozoa, you can show by fluorescence microscopy and electron microscopy that they have some capacity to damage these sperm. They actually make them die. And also that they will stick to various parts of the sperm. But what wasn't clear, it's all very well doing these experiments with lots of chlamydia in the lab. Is this really true in clinical samples? And so we were very lucky that Adam Brooks Hospital had spent 6 years collecting samples from various patients. And we did some molecular diagnostics. So we were able to actually look at how many of these patients were positive for chlamydia, how many were positive for other genital infection causing bacteria, and some of which are also doubly infected. And we found that 16% of these were actually positive either as a double infection or as a single infection for chlamydia, both in the urethral swab samples and in the semen samples. So this gave us a repository of samples which we could conclusively or conclusively show were definitely either chlamydially only infected or a mixed infection, where we could ask questions using some of our imaging techniques. So here we see this is now sperm which have been stained for fluorescence microscopy instead of intact large eukaryotic cells, which in itself is difficult to do, and so the signals that you see are actually dimmer. What I hope you can see is that there are green structures attached to the head of the sperm, which is in blue here. So this is an anti-chlamydia antibody lighting up an elementary body, which is stuck to this sperm head. We see one down here that's much harder to see on the mid piece. We see another one on the head here, and another one on the tail. And if we look across our panel of infectious patients, we can add up the frequency of these observations, which seems to be proportionate to the size of the membrane. So more chlamydia stick to tails because tails are larger than heads. There doesn't seem to be any preference for heads over tails. And yet 7% of the total number of sperm in these patient samples were associated with chlamydia. And so we wanted to look at this in more detail. So we then tried to prepare these sperm for electron microscopy, which turned out to be quite a headache. We eventually managed it, and we were able to look at these patient samples again and show, quite to our surprise, that not only were bacteria obvious in proximity to the membranes, but particularly in the sperm heads of a number of independent infected patients, we saw the generation of some sort of compartment within the sperm head, which seemed to be, and we can now show to be through immunogold labeling, as we did with the needle structure, to be chlamydia, which seemed to be enveloped inside this sperm head. And indeed, some of them seem to be enveloped totally in a compartment, perhaps reminiscent of an inclusion-like structure. Here we see some sort of invagination in a flask-like structure into the head. There certainly seems to be a boundary of sorts to this structure. So this leads to the question, should we really be looking more at men? Can chlamydia at least be carried or directly influence male fertility as well as female fertility? And this is really the first time in any patient study where these kind of interactions have been visualized. So in terms of our fundamental research, we're now pushing forward using our cell models to try and find small molecules which are alternatives to antibiotics, which may be able to interfere with the generation of these inclusions. So here we have an inclusion again sitting down here. And I've shown you on the top just one example of a compound, whereas if we treat these infected cells, we see that the bacteria, which are quite small down here along the boundary membrane, swell up. And through biochemical assays, we can show these chlamydia are actually dead, and that this inclusion is then expelled from the infected cell. And we can show that the reason for this is that the cell begins to sense the presence of the bacteria triggered by the molecule that we've added to this infected sample. So we see now that this compartment here, but not the normal inclusion, is quite clearly stained with a marker consistent with the trash dump that we saw in that first slide, the lysosome, which contains enzymes that are responsible for degrading elements inside eukaryotic cells. So we hope through these assays that we might be able to investigate new compounds that may illuminate new avenues, at least, to potential new treatments in the future. In terms of managing the disease worldwide, at the moment there's quite a good system in place in the third world called the SAFE strategy, which relies on a combination of surgery, antibiotics, increased hygiene, and access to clean water and sanitation, called the SAFE strategy. Obviously, this is to some extent dependent on antibiotics and doesn't prevent patient reinfection unless surgery has taken place, but is at least constraining chlamydial incidence to particular geographical areas such that it can be isolated and then hopefully at least the incidence decreased by the use of antibiotics, which Pfizer have denoted at the moment worldwide to try and prevent these infections. So whilst we can see the need for a genital vaccine, it's possible just through the increase in hygiene and living standards, as we might imagine happened in medieval England and during the Industrial Revolution, we can actually use these kind of approaches to knock down at least the incidence of chlamydial infection and trachoma and blindness in third world Africa. And this is something that the London School of Hygiene and Tropical Medicine are heavily involved in monitoring and investigating. So the final question is the balance of power between screening and vaccination. So the NHS and the government invested a large amount of money back in 2009 into a national chlamydia screening programme. You may have seen the adverts on the tube and on the TV. And we've just had the outcome of that, which was somewhat disappointing, that the national screening programme didn't actually lead to any significant fall in the infection rates. It told us a lot about infection rates, but it didn't really result in less people being infected. So the conclusion was that actually it's very hard to change teenagers' behaviour, and so perhaps we should be diverting the funding rather than continuing in this screening towards the development of a vaccine. And the flagship example of this is the recent success and deployment of the human papillomavirus vaccine against cervical cancer in teenagers. And it's hoped through our work and others in the field that eventually we might, and we are beginning to understand, how to better produce these vaccines against chlamydia in terms of understanding the immunology of the infections and these proteins that come through and are deployed into the cells, because these will give us clues about which are the so-called protective and preventative antigens, the protein that the body recognizes that signal the bacterial infection that can be developed subsequently into the vaccines. So what I've hoped to show you is that certainly for the last 15 years it's been an exciting time to be a microbiologist, and I'm sure that will be continuing, because we've moved from a world of classification through molecular genetics into understanding genomes of organisms. I've shown you how cellular microbiology is now informing our experiments. And I'm sure in the coming years that structural biology will also begin to contribute to our understanding of bacterial disease and new ways to inhibit them. And if you're interested in hearing more about this, I'd point you towards my colleagues and myself who teach on our various courses in microbiology and health and disease here at Birkbeck. So with that, it's very important to acknowledge the people who work in the team and our collaborators and our funders. So I'm very lucky to have built the team in my last 4 years here, to have some very talented postdoctoral researchers and PhD students working in my lab, complemented by a number of master's and bachelor's project students. I have a very productive collaboration with Professor Helen Sable and her postdoc Dan Clare. Dr. Hamid Jalal and Christopher Khan are the collaborators in Adam Brooks and at the Health Protection Agency, and these are other collaborators on ongoing projects I haven't described this evening. As I said, my position and my work is funded by the Royal Society and is augmented by studentships from the BBSRC, the Biological and Biotechnological Sciences Research Council, and the Medical Research Council, and also a small charity, the Infertility Research Trust, has been very generous in supporting the work on male factor infertility. So with that, I'm happy to take any questions, and thank you very much.
Questions for Richard? No? Okay, well, thank you once again for coming, and we have 2 more talks tomorrow at 4 o'clock. Professor Bonnie Wallace will be talking about sodium receptors and pain, and at 6 o'clock we have understanding medical genetics and diagnosis from a psychological point of view by Professor Jonathan Smith. Thank you once again for coming.